So Professor Mahato is a professor and chairman at the Department of Pharmaceutical Sciences, University of Nebraska Medical Center, Omaha. He was a professor at the University of Tennessee Health Science Center, research assistant professor at the University of Utah with Professor Sung Wan Kim, senior scientist at Gene Medicine INC, and as a postdoctoral fellow at the University of Southern California in Los Angeles with Professor Vincent Lee, Washington. He has also worked in Kyoto University with Professor Mitsuru Hashida. He received his Ph.D. in drug delivery from the University of Strathclyde, Great Britain, and B.S. from China Pharmaceutical University, Nanjing. He has published 160 peer-reviewed papers, 24 book chapters, held 3 US patents, and has edited, written 9 books and 11 journal issues with a total citations of more than 11,000 and has an H index of 62. He is an associate editor of the Journal of Neuroimmune Pharmacology, was a feature editor of Pharmaceutical Research from 2006 to 2013, and is an editorial board member of 8 journals. He is a CRS Fellow 2011, AAPS Fellow 2010, is a permanent member of BTSS NIH Study section, and ASGCT Scientific Advisor, non-viral vectors from 2006 to 2009. He is applying sound principles in pharmaceutical sciences in the context of latest advances in life and material sciences to solve the challenging drug delivery problems in therapeutics. His areas of research include drug delivery and targeting of small molecules, microRNA, and genes using novel polymeric and lipidic carriers for treating cancer with a focus on pancreatic cancer, prostate cancer, and melanoma, liver fibrosis, and diabetes. And with this introduction, I invite Professor Mahato to deliver his talk and give his insight in the field. Now over to you, Professor Mahato.
Yeah, first of all, thank you, Deepak, for your nice introduction, and also my thanks to your department chair, Dr. Jadhav, and all students and faculty for finding time to attend and listen to my presentation. And especially for me, it really gives me a great pleasure because for some of you may not know, but I was really born in Nepal. You might have heard Janakpur, which is it's Mithila, right? And then from there I went to China. So naturally I can speak Hindi, I can speak Chinese, I can speak Japanese, you name it. But with the interest of time, I really want to share with you what I learned in this journey. And so one topic I really want to share with you about how can we really combine small molecules and nucleic acid drugs for example, microRNA, which is non-coding RNA for treatment of different diseases. But for this presentation, I have focused mainly on pancreatic cancer. So first, let me tell you why I'm so interested and why especially young people there should be interested, because the work we do, basically we really apply nanotechnology. As you know, nanotechnology you can use in anything. It's a buzzword now. You can use, but for medicine, especially in pharmaceutical science, we can really use it wisely for prevention of the disease, for the treatment of diseases. You can use for diagnosis. And really the overall goal of our purpose really is very simple. Because, we study pharmaceutical science. Some you may say that really I really study to buy a good car. Some may be dreaming to have a big mansion. But really ultimate goal is really how can we pharmaceutical scientists improve the quality of life and lifespan of a patient. And this is our ultimate goal, and that's what we are trying to do. And we believe we can utilize nanomedicine approaches to achieve these goals. So my lab is really very diverse, simply because I have very diverse expertise. Certainly, I always believe in designing career, like in terms of polymers in the past. Even now, we talk about lipids. Sometimes we design peptide. When I say peptide, this career means design means we try to synthesize in the lab. and for different purposes. But at the same time, I always believe that really disease can be treated better if we can treat at a molecular level. So therefore, you could really also use gene expression systems to express or silence certain genes. And for this, I've done extensive work to really design different expression cassette to express genes like hairpin RNA or even microRNA. You can do that. And ultimately, by training, we are pharmaceutical scientists. So naturally, we do the formulation and we assess in vitro as well in animal model in terms of cell culture work, pharmacokinetic, pharmacodynamics. On and off, we also use pharmacogenomics because there is a patient-to-patient variation of the same therapeutics have different response and why it is. So that's we also try to do it. And we do this really in three different disease model. One is liver diseases, and one is in diabetes, and also in cancer. So today I will talk more about cancer. And in each case, one of the key issues probably many students there will be interested because our goal is really to deliver drugs. And drug, when I say the drug can be anything, a small molecule, macromolecules. And in each case, really you can deliver in two ways by using particulate systems, or you can use by conjugation approach, so-called micromolecular systems. So in particulate system, my lab is really involved with both, like liposomes, our nanoparticle, of course, our micelle, and each has own advantage, disadvantage. And with the interest of time, I'm really leaving up to you to learn about this. But occasionally, we try to really conjugate our drugs or nucleic acids to certain polymers or lipids using pH sensitive or some kind of cleavable spacer. And this is really for two reasons. One, to really simply increase their stability, but also to increase the target specificity. And this all really we have richly expressed in discussed in different publication from my lab, and these are some of the examples of the books or the theme issues. Some I've listed, some not. And I would really encourage, especially students and young faculty, to really to find some time to look into this. You may find something of interest, and I'll be happy to even send you a copy if necessary. But today, really, I'm trying to share with you about the tumor. And the tumor, when we talk about the tumor, as you know, there are different types of tumors. And I'm trying to tell you about pancreatic tumor. But here, the tumor, what you will have is you will have different cell types. And among them, of course, there will be tumor cells. There will be also macrophages. There will be extracellular matrix. And of course, tumor, you will have also hypoxie conditions. And there will be high interstitial pressure, so which will retard the distribution of drug in the tumor. And quite often, especially in case of pancreatic tumor, there will be also desmoplasia because why this is so important, because desmoplastic tumor does not really allow efficient distribution of drug to the pancreatic tumor. So therefore, this is a big issue and need to be addressed. And there we believe our nanotechnology approach can be used to really address this. And of course, we have to really address a lot of biological cancer biology issue. And if you're interested, you're welcome to really visit our special issue to address some of these concepts. And first, up to really 2006 or so, I was really more, 2007, you can say, I was more interested in nucleic acids. Basically, I was working on genes like virus and non-viral gene therapy. I was working a small, siRNA, but I really, one student visited from from Kentucky. This guy is here. His name was Michael Dangwa, and he really wanted me to really work on work on a small molecule. Michael Dangwa now is a department chair in Chicago, and he really wanted to begin the work with the polymeric micelles. And this is the benefit he shared with me to really win me back, because I was really very reluctant to work on this. And basically, you can use the micelle. First, is very simple, as you know, the surfactants like properties. It can is small in size. It can have high drug loading. You can really control the release of the drug, our site specificity, and so on. So therefore, I got interested, and also most importantly, from beginning, I was really less interested in using commercial polymer, and therefore Michael really came up with the idea to modify the polymer by introducing polycarbonate unit into this. And this really helped two things. First is increase the drug loading, and also the degradation of carbonate unit really is not acidic. And therefore, it became very attractive, and we did some work on that. And students, if you're interested, really visit that. But of course, in between many, many things. And of course, we did a lot of work initially with a small interfering RNA. And I asked my former student, Dr. Puncham, who is now full professor at University of Missouri, Kansas City. He's very rich, a lot of grants. So Kun really tried to focus on siRNA. But at the same time, I had a postdoc who joined in 2008 from China. And he was very much interested really to initiate microRNA, which is non-coding RNA. And he is now a really full professor at Tongji University in Shanghai. And that's why we try to compare why should we really switch from one area to another, why from siRNA to microRNA. And he impressed me because the number of publications, as you could see in this slide, is significantly higher and very rapid increase for microRNA, whereas the number of publications for siRNA beginning to slow down. So I thought it's a good area to get started. And then, of course, with some time, there was another postdoc who is, I think, now in India, Dr. Saurav Singh. I think he was working in Novartis, India. I don't know where he is now. And at the same time, I visited at that time, I think it's in India, and I met Deepak and Anupa, and at least I win back Deepak to come to my lab as a visiting student. And the reason was very simple, because I like the guy, and I thought he should really spend some time with me. So Deepak and Anupa joined, and they really began to come up with the idea. I'm trying to make it short, really to combine a small molecule with microRNA. And basically, they were interested in gemcitabine. And as you know, in this slide, I very richly explained the reason. And with the interest of time, I just want to tell you a couple of things which draw my attention. One, because whether it's gemcitabine or any other drug, if you give high dose, it will really lead to systemic toxicity and also really cause chemoresistance. And therefore, we thought we can combine microRNA, which can really inhibit chemoresistance, and in return can chemosensitize the cancer and cancer cells. And then later also Birendra Kumar, he got involved. He was a student at that time. Now he's an instructor. He should be promoted to faculty here. And they begin to work on that. So, but only my request to Deepak at that time was really, I want to deliver both microRNA and the gemcitabine together as a single formulation. So that's why Deepak really introduced this cationic chain into this polymer, which will allow the ionic interaction with the microRNA, and also he conjugated gemcitabine to the polymer. So basically, it formed the polymer drug conjugate, and because of ionic interaction, microRNA got protected because it got encapsulated in the core of the micelle. Because once you ionic interaction, they basically insolubilize microRNA. So with this concept in mind, they did a lot of work, and this is the kind of design element. And of course, they have a very nice publication biomaterial, and you are more than welcome to read on that. And but I also was really interested to see really does it really help bioconjugation in terms of biodistribution, and if so, really it can also does it help in terms of really inhibit the metabolism or slow down the metabolism of gemcitabine. Here, gemcitabine is abbreviated GM, and there was the Krishna Patel. He was from Nepal, but he has done Ph.D. From South Korea and postdoc in Japan. And he joined my lab, and he really did a biodistribution in orthotopic model. And he clearly saw that first, of course, it can really increase the drug distribution, and also at the same time really decrease the metabolic conversion of gemcitabine. And the next slide, as you could see it affect in the tumor in the ear. The distribution to the tumor was increased by 2.5-fold after systemic administration. And keep in mind, these are not sulky tumors, they are orthotopic tumor. So we felt, and of course measured by, so it was a good mode and move. Then after that, of course, we did a lot of work. We measured the tumor, but we realized there's some decrease in tumor size, but really still tumor decrease inhibition of tumor growth is not as good as we like. And therefore, I introduced another fellow. He is Gautam Mandal. He is now in another university. And Gautam, he was a postdoctoral fellow. And what he did is he introduced targeting. So he basically attached targeting ligand. Here he used antibodies to target EGFR receptor, which is overexpressed in pancreatic tumor or any tumor per se. And he saw he demonstrated that, of course, it first increased the distribution, and this conjugation can really inhibit the inhibit the tumor growth. And apart from antibodies, he also used GE11 peptide, which also targets EGFR receptor, and he saw fairly good benefit in terms of inhibiting tumor growth. Then now question I wanted to tell you all for educational sake, especially for our students, why we are really interested in microRNA. Why not bringing another drug? And this was really discussed. First, we discussed in one editorial by my former student, Michael Dangwa, which, as I told you now, is a department chair. So he, at that time, really explained the microRNA has very unique properties. Because first, microRNA, as you know, it really affects EMT, epithelial to mesenchymal transition. And that EMT transition, as you know, quite often the cancer stem cells our cancer stem cell phenotype quite often being affected or converted also due to the aberrant expression of microRNA. And other thing which we also realize that microRNA really deeply involved in chemoresistance. And therefore we felt that if we could target not only the bulk tumor, but also cancer stem cells, CSC, cancer stem cell, and EMT cells, then will be good because they are chemo-resistant cells and have metastatic potential and express a high level of hedgehog ligand, which I will explain you later. Also we clearly realize that this regulation of microRNA is really associated with tumor initiation, differentiation, EMT phenotypes, like cancer stem cell proliferation, etc. And therefore, use of microRNA becomes very, very important. Now, question is which microRNA will use? So, a lot of people in the pharmaceutical field, basically, by nature, we are people try to mix up, right? Mixing to prepare compounding. But from beginning, I was really reluctant to do that. So, we did a lot of profiling. So, we did profile the microRNA expression in different cell types, also patient tissue. And we realized that some key microRNA are upregulated and downregulated, and then we use bioinformatics to really decide their target genes. So here, one microRNA we got interested was microRNA 205. And the reason we were interested, because as you could see it, when we did bioinformatics with software, it's free, done mainly by students. Really, I don't even know how they do it, but they really came up that this microRNA 205 affects a lot of things. It really affects drug cancer cell invasion, migration, metastasis, then the radio resistance, chemo resistance, you name it. So therefore, it became a very important target microRNA. And this microRNA, as I told you, it is downregulated in the pancreatic cancer, right? So therefore, we use microRNA mimic. And this first was really done by Saurabh, even before Deepak began working on this. And he clearly told us that when we use resistant, gemcitabine resistance cells and combine and then treat with microRNA 205, then it will chemosensitize the cells, and that will lead the killing of cells with increase of gemcitabine concentration. So clearly so that, and also the sort of really clearly showed that microRNA 205 has significant effect on certain genes which are directly associated with chemoresistant, like OCT34 cells, OCT34, like CD44, like TOB, especially TOB. TOB is directly related to chemoresistance. So there was a very good rationale behind of using microRNA 205 in combination with gemcitabine. And this, of course, this is the formulation. I think Anupma and Deepak did it, and they confirmed that microRNA need to be dissociated. And for, as you know, microRNA, you cannot see it, so they did the gel electrophoresis to confirm that it can dissociate, especially in presence of DTT, and suggesting that really you can formulate both at the same time. And then, of course, they showed even in animal model that their combination indeed will have some benefit. So before but one problem with here was, as you know, that a couple of issues I wanted to share. First, gemcitabine is a water-soluble drug, is rapidly metabolized, and since it's a water-soluble drug, we have to really conjugate to the polymer. So then Birendra got involved, and he suggested, Dr. Mahato, rather than using gemcitabine, we should use Hedgehog inhibitors, which he was working with liver fibrosis, because Hedgehog inhibitor is insoluble in water. So basically, you could really encapsulate. So you don't need to really conjugate to the polymer. And also he suggested rather than using 205, we should use LEAD7B, another microRNA, because that is probably more potent than 205. And that's what he thought. And so again, we did, and the reason we selected Hedgehog inhibitors simply because, whenever you have inflammation, whether it's cancer, whether liver injury, there will be upregulation of a hedgehog ligand, like a sonic hedgehog, like Glee, like Smoothen, and this we really saw Western blot as well as real-time PCR, and therefore inhibition of this really lead to a benefit. And some of you may remember that in fact there is a science paper that the clinical trial with Hedgehog inhibitor failed. And the reason they failed, because they use a free drug, and free drug, if you use a very high dose, then naturally it will cause toxicity and so on. So that's why we thought we should formulate this they had also used a single drug, but in our case we used with miRNA late 7B, and then we did show that indeed this is a beneficial to do that. And of course we measure a lot of other genes, and we check with the different pancreatic cell line to really convince that really late 7B is down-regulated, and therefore its combination of combination with GDC, which is a hedgehog inhibitor, will be beneficial. So we did, of course, a lot of work there, and we could easily formulate this together. And the presence of microRNA had little effect on GDC release. And of course, we check in animal model, and as usual, we measure tumor size and so on, and saw that tumor growth is slowed down. But as you remember, gemcitabine is a commercial product, number one. And the gemcitabine, another issue is still cause resistance, means after repeated use, it doesn't work well. And the binding efficiency to mutated smoothing is very, very poor. So because of this reason, gemcitabine is not as good as being promoted. So we thought maybe we should do something about this, right? So then I had, being a department chair, you have one benefit because you know the people who really need some work. So I met Yu Xiangdong, who is a research faculty, and also is from Nanjing, so where I spent some time. So I asked Yu Xiang that, Yu Xiang, I'm working with hedgehog inhibitor. Can you help me design this with some modification? And I'm not really pure chemist, but I draw something and I said, this is this, and can you really make some changes? Then Yu Xiangdong really designed a lot of analog of GDC. Some I'm not showing you simply because Deepak told me that you want to put in YouTube or something. So unpublished thing I didn't want to share. But MDV5 I'm sharing with you because this is already published, and in fact is now being manufactured by one of the company in US. Of course, I don't have patent right because I published without that, but the company, in honor of our contribution, they still kept our name MDV5 in their product. So that's good. So we did it, we modified this, and compared to to GDC, naturally it is much more effective in terms of inhibiting hedgehog ligand. And most of the Western blot is done by really Bharti, which is from Haryana, I guess, or somewhere from Punjab, and he's a graduate student here. And he worked closely with Virender, and you could see that whether it is on the silencing or the IC50 value is much better than GDC. But I want to really draw your attention. It's not so common.
Good morning, everyone. Am I audible or some echo is coming?
Oh, echo is coming? I don't know. I am not keeping anything on. Everything
This is fine, Professor Mahato. This is fine.
From our side, maybe one of you might have kept your speaker on, but really not from here.
That's okay. That's okay, Professor.
Yeah. So let's move on. So what we did, we designed a lot of them, and I show you a couple of data. And one data, just to, of course, we did a lot of things. Most of the things are published. So interested student or faculty can really kind of also really check our literature. But one thing I wanted to tell you that MDV5, which is a hedgehog inhibitor analog of GDC, naturally is much more effective than GDC alone. So that's the main message. And we published a scientific report for cancer. Then we published diagnostic for liver fibrosis and so on. And I don't have all in my head, but you could really dig out our presentation. But I want to really share with you today's topic, which is something different with the time I have. And I told you why I'm trying to show this to you. The first thing is Sorry,
I missed that. Could you say it again?
Yeah, I'm sorry. So first is the redox-sensitive.
I'm not sure I'm here.
Oh my God. Yeah, I apologize. So redox-sensitive polymeric system, we thought we should use it. And also we thought we should utilize hypoxia. Because as you know, pancreatic cancer quite often undergo hypoxia, right? So the hypoxic condition is one of the key issue, and another is of course desmoplastia. So this work really initiated by Vinod. He is now at Pittsburgh, and then just like Deepak, when I went to India, I stole Deepak back to US, brought him US. Same way when I went to China, which I go almost every year, I met Xiaofei, this girl, and her husband, at that time boyfriend. So they were graduate students. So I asked them, okay, they should come to US as a visiting student. They joined my lab as a visiting student, and now of course they are visiting a scholar. They finished PhD, and so she really worked basically on this project. And then this is the Feng Lin, my former graduate student. He's very good polymer chemist, and he helped us design the redox-sensitive polymer to deliver this. And I'll tell you as we go around. So question is first, why should we use 519C microRNA? Why not microRNA 205 or let's 7V? The reason is very simple, because microRNA 519 has some of the properties very interesting. So first, of course, just like microRNA 205, microRNA 519 is down-regulated in tumor, and also there and the microRNA 519 expression is severely decreased, is reduced in cancers, pancreatic cancer cell line. And like here, resident Miyapaka 2 cell line is certainly very, very low. Capans 1 has also very low expression. And another thing we found, as you could see, this was confirmed with stone blot as well as real-time PCR. So we felt that we can use this, their combination. And I'll tell you why we think so in the next slide. So here, as you could see, first, when we created under hypoxic condition, right, and we use a scrambled microRNA, and we use microRNA 519, we clearly saw that microRNA 519 has beneficial effect in terms of inhibiting the migration of the cells under hypoxic condition, as well as under desmoplastic condition. For desmoplasia, to create desmoplasia, we mix our cells with hepatic stellate cells, which is the cells present in pancreatic, the human pancreatic stellate cells. So pancreatic stellate cells present in pancreas, and these are the cells really produce ECM, excess amount of ECM, including collagen, and that cause desmoplasia. So that's why we mix the cells with this pancreatic stellate cells, and under hypoxic condition, as you could see it, a scrambled microRNA has no benefit, but when you use microRNA 519C, this is a benefit. This is a really very extensive work. I'm just sharing with you a few slides. This will be out next week. I think November 11 in Science Advances. So students, if you want to read it, you could do it, because I'm really not allowed to put the citation before November 11. Is advised by the journal, but you will have access from November 11. So the other thing is the other reason we are interested in microRNA 519, because microRNA 519 inhibits glucose metabolism to really inhibit gemcitabine resistance to pancreatic cancer cells under hypoxic condition. And why I bring metabolism? Metabolism is also very, very important, as you know, because of this. In fact, I've asked Dr. Mittal, Anupam Mittal, to really work with me for one special issue we are working, so-called diabetes-associated fibrosis. So she's trying to create one special article on that, because diabetes, in cancer is very, very important, and so is in case of fibrosis. And when you talk about diabetes, then naturally metabolism, glucose metabolism is very, very important. And that's what we wanted to do. So as you could see it, under hypoxia condition, especially under hypoxia condition, microRNA 215 really inhibited GLUT1 expression, whereas the scrambled microRNA has little effect. And another good thing is, and I apologize for this slide, we mixed some of the writing, but basically what you can see is the DCTP level was also significantly decreased by the treatment of microRNA at 519C, but with the scrambled microRNA had little effect. This is under hypoxic condition. And the other benefit, as you could see it, when we and we also saw that really it affects on many other important genes related to glucose metabolism and so on, and also it has effect on deoxygenosis, what you call this uptake, deoxyglucose uptake. As you could see it under hypoxic condition, this microRNA 519C significantly decreases its uptake. And this has effect because this is competing the uptake by gemcitabine. So it is, and that's why this becomes very, very important to really to use microRNA 519C to really enhance chemoresistance and also inhibit so-called this glucose metabolism. So with the interest of time, the other thing you could see it, and we clearly saw that microRNA 519C and gemcitabine have synergistic effect in terms of inhibiting pancreatic cancer cell proliferation as well, and also in promoting apoptosis. So I have really excluded many slides related to apoptosis, but I do want to share with you one, as you could see on the left side, as we combine microRNA with gemcitabine at different concentration, as you could see, the combination has significant effect in inhibiting microRNA, inhibiting cell viability. I apologize for this, this statistics, all symbols mix up. And also we created 3D model, and 3D model, as you could see it here, so like tumor fluid we created, and tumor fluid is the control non-treated tumor fluid. This is the tumor fluid treated with microRNA 519C alone, very few cell death, apoptotic cells with red cells, red dot represent the cells undergoing apoptosis or dead cells, and this is with gemcitabine. But with the combination, as you could see, combination of with the gemcitabine and microRNA 519C, as you could see, in tumor fluids, majority of cells are really undergoing apoptosis. So you could see in red color, and that clearly, and of course we also did like cell cycle, a lot of apoptotic genes we measure, and we saw similar effect. The other thing I wanted to share a little bit, changing the gear, like microRNA, right? So first, the challenge is selection of the right microRNA. And as I said, depending on the investigator, some don't want to waste time, so they just buy from the company. And some really do the profiling, right? But the other key thing is, just 1 second, because I want to see the time. Yeah. So the other key thing is, so when you use the suppose even if you select microRNA, the right microRNA, is it done deal? Answer is no, because microRNA is a nucleic acids, right? And especially single-stranded non-coding RNA, fairly unstable, very, very unstable, especially in vivo situation. And therefore, the challenge is how to do the backbone modification, so-called chemical modification of this microRNA, which should really not have any adverse effect in terms of its hybridization or so-called its functionality, right? But it should increase the stability. So we really did a lot of backbone modification. And I'm showing you one with the interest of time. So one modification we did it, basically, we introduced two methoxy, and also partially phosphorothioylation. So these two modifications in the same backbone had a lot of benefit. First, it could significantly increase the stability. As you could see it, when we incubated in the serum, microRNA without modification degraded very quickly within 6 hours, almost all degraded. But with modified microRNA, even in 24 hours, most of them remain intact. And now, of course, some students may ask, why not simply using so-called PS or fully phosphorylated microRNA? The reason is when you use fully phosphorylated microRNA, which I did long time back as oligonucleotides when I was still in Japan, that they really increase significant binding to the protein and plasma protein, and also it affects hybridization, and that's why it's not good to have fully phosphorylation. So that's why we did this kind of modification. And this modification, another benefit was this really also in fact to some to have slightly benefit in terms of increasing the efficacy of microRNA. And most importantly, it really decreased the immunogenicity associated with microRNA. Because microRNA, you don't want to use a microRNA should really cause any immune reaction, and which you could see here. And let me have just 1 or 2 slides. Then, of course, we did the formulation and characterize in terms of particle size, like surface morphology, atomic force microscopy, drug release, etc., etc. We all did it. And then, we check in animal model. We saw that indeed redox sensitivity is important, and it has really decreased the tumor size significantly. And this is important because when you see in this slide that our polymer indeed it is pH sensitive also, so it can control the release or maximize the release of microRNA after internalization to the tumor. So finally, I want to escape this side. Finally, I want to end this talk. All this work, what I really want to tell students and some faculty alike, collaborative effort is very, very important here. So when I meant collaboration, sometimes we do collaboration for the sake of collaboration. And that I'm totally against, because don't do collaboration simply because he's your department chair or he's your dean. Again, I'm not against Dr. Jadhav, don't get me wrong, but do collaboration only if it is a genuine reason. For example, like Pixar, when you combine like toys story, right? Music and engineering combined together, right? Then you have big shift in terms of improvement, and this is what is needed, because what young minds should think of is 10x or beyond really improvement when you do this kind of method, rather than really one-fold increase or 0.5-fold increase. That is not really going to do anything. So therefore, I do encourage you wise collaboration. And finally, what I would suggest, the era of single using monotherapy is really gone. So quite often you have to do the combination therapy, and I do encourage you to consider microRNA. And when you do that, really don't simply buy microRNA. Try to find out which microRNA you need to do. You may have to do microRNA profiling. You may have to learn a little bit RNA sequencing so that you really pinpoint the exact microRNA. And I would prefer to begin with some patient sample, and then you do testing, and then if necessary, again you reverse back to make necessary changes in your formulation and so on. So this all work really done by, not by me, mostly done by a lot of people. Some of the names are missing, seems they didn't get time to really acknowledge all. But I also like to thank the funding agency, of course, university, and NIH, Department of Defense, Pancreatic Cancer Center, etc. With that, I rest my case, and now I'll be more than happy to answer any question you may have. How to remove, and Brenda? Just escape, right? No. Can I say? Yeah, so. Yeah, so can we make the... You have any question, or do you think I ran out of time? Or?
Yeah, we can have quick questions if anybody have.
Good morning, sir.
Okay.
Yes,
Dr. Mahato, I have a quick question.
Yeah, please tell me, yeah.
Yeah, it was wonderful talk. I'm very happy that I could listen. And so just one quick question about technical things, that is it always required to prove the synergism by some techniques like statistical techniques like Chow-Thalai methods or something, or just cell viability like significant inhibition of the cells, proliferative pathways are enough?
No, actually, you have to do many different ways to prove there is a synergistic effect or therapeutic effect. So I just showed you one cell viability, but you have to also do like different marker like colony formation assay and so on. So there's a lot of technique you have to do, but in the presentation, naturally, I can't show you all because I just simply wanted to make a story, but you have to do all other technique. But other really, I want for you to think of, rather than killing a lot of times in vitro dealing with cells, I would suggest to really go in animal model and the right animal model, so that you will have better impact of your study. Did I answer your question or not? Yeah.
Yeah, go ahead, sir.
So it means you have to do more than one technique, like mRNA level, like Western blot level, immunohistochemistry, and also you have to use a statistical analysis. So most of we do apply statistical analysis, but honestly, now I reached the phase I don't have time to prepare those things, so I just do it very fast. But people in the lab, they certainly apply all the statistics because this is absolute requirement. Because as you know, NIH now is very serious about reproducibility of the data, and that's why statistical analysis become very, very important.
Right. Okay. Thank you. Thank you.
Yeah. Any other question?
Professor Mahato, I have a quick question.
Yes, please.
Whenever we are talking about combination therapy, generally we target the same cancer cell, right? From different mechanism. Now, what is your suggestion regarding finding out those targets, that this pathway I can target along with that one? So
Yeah. My suggestion is, number one, don't use the two different combinations for the same target. Even if you use the same disease, tumor or maybe other disease, right? But that can be, I give you example, like for example, like pancreatic cancer, right? So one can be chemoresistance, but can be also age-old issue, right? Can be desmoplasia, right? So if you use more than a couple of targets, then the benefit will be better, So that's, and another thing is, try to target not only bulk tumor, but quite often tumor initiating cells, because that's very, very important. But in some cases, for example, if you do like liver, like cancer, in that case also inflammation become very important. So depending on the disease types, you may have to choose wisely. And that's why cancer biology or any disease pathophysiology understanding is super very, very important. And that's why I, especially in my lab, I require a student to really invest some time to understand cancer biology or if other disease pathways, so that they should know different signaling pathway, and that way they will select the right pathway, combination therapy, because you don't want additive effect, right? You want synergistic effect, right? Yeah.
Synergistic effect. Thank you so much, sir. Thank you.
Yeah. You're welcome. Yeah. If any other question, if not, then you are more than welcome to write to me. I will be happy to answer any question if you have, and certainly it was my pleasure to talking to you all. And Deepak, anything else you want? If not, then
thank you. Thank you, Professor Mahato, for giving this insightful talk and motivating the students and young faculties like us to look deeper into the field. Thank you for spending your time for that talk.
Yeah, that's most important, because especially for young faculty, I'm not old, but still I feel a little bit older than some of you. If you're younger faculty, don't go for quick return, you invest something to go deeper, and that's very important. That's something I made a mistake, because I tried to have a quick return, and in the process I become very superficial. So I would strongly recommend to go a little bit deeper, and publish in a high impact journal, which again, most of us, including myself, are failing to do so. Yeah, thank you.
Yeah, thank you, Dr. Mahato.